of Biophysics, University Linz, and the title of the talk is The Multiple Roles of Ori and Terminus in CRAC Channel Function.
Okay, thanks for the introduction and welcome to my talk on two main key players in the calcium signaling cascade. We are basically interested in two proteins, stem Ori, but first of all, what is calcium useful for? Calcium has many essential, is relevant in many essential steps in the throughout cell's life, so it controls cell growth, cell development, but also cell death. Further, it's also involved in processes such as exocytosis, muscle contraction, cell growth, and gene regulation. And one main calcium pathway into the cell is the so-called store-operated calcium ion channel or store-operated calcium entry pathway. How are these store-operated calcium ion channels activated? For that, a ligand has to bind to a receptor in the membrane. Here we specifically talk about immune cells or mast cells. And the signaling cascade is activated, which leads to the production, among other substances, to inositol triphosphate, IP3. IP3 binds to an IP3 receptor in the endoplasmatic reticulum. The endoplasmatic reticulum forms as calcium stores in the plasma membrane. And when IP3 binds there, it undergoes a conformational change, this receptor, and it leads to the release of calcium from the endoplasmatic reticulum, as you see it here. And this leads to the activation of calcium from outside the cell, as you see it here. So this store-operated calcium entry requires the activation of the calcium stores, so the depletion of calcium from the endoplasmatic reticulum. And this process has been known for a very long time, so several decades, but the two proteins which are relevant therefore are only known now for 12 years. And there is one protein which sits in the ER membrane. It's the sensor protein. And another protein sits in the plasma membrane. It's the channel protein, the calcium ion channel. So the sensor protein is the stem protein, which you see here. It's a single transmembrane protein localized in the ER lumen. And it has one EF hand, which senses as a calcium, which functions as a calcium sensor, so it can sense calcium in the ER. And it has a very long cytosolic tail, which functions as a main binding domain for this coupling partner in the membrane. So this coupling partner in the membrane is a calcium ion channel. It's the Ori ion channel, as you see it here.